Anyway, let me start by thanking Brain Foundation for the opportunity to talk about transcranial photobiomodulation. This is something I've been doing for 25 years. So let me start by saying the object of my talk is not so much to get into any one thing about these in any detail, but to go through it to motivate and hopefully get you interested in that technology so that whenever you do your research, whenever you look at clinical studies, whenever you look at therapeutics, it comes to your mind. So the first thing is, what is it? So if you look it up on Google, you're going to find these long-winded definitions. To me, it's just simply the use of near-infrared light that is delivered transcutaneously to the brain to modulate the physiology of the brain. So the first thing that you ask yourself with something like that is, keep doing that, huh? How do you dose it? So most of you guys, maybe I'm speaking out of turn there a little bit, but you're probably very familiar with pharmacological applications or therapeutics. So you're talking about dosings in one way that is very different from the way I would think of dosing. So how do we dose light? And the reality is that it is complicated. There are just too many parameters to specify a dose and to actually try to test all these parameters in clinical studies. So you have to make certain assumptions and you have to pare down this infinite space into something that's falsifiable. So here there are two things that I would say about dosing that I think if you ever get back into the subject or you're interested in the subject are critically relevant. So the first one is wavelength. So what's the wavelength? Probably we don't have to get into that too much. I'm sure everybody knows is electromagnetic waves propagate through space as waves. The wavelength is the distance between a peak and a peak or a thermometer. Now, I think of wavelength, maybe the analogy here, taking some liberties, as perhaps the drug that you're using for your therapy. The other critical parameter here, and by the way, the specification for wavelength is nanometers or micrometers, so if you're reading a paper about this should show up. The other thing that's important is the fluence. So fluence is the energy delivered to the tissue per unit area. And it's specified in joules per square centimeter, or more appropriately, in watts per square centimeter times the exposure time. Again, taking that same analogy with the liberties, this is more like the drug's concentration in the cells. So now you have the drug and you have the drug concentration. You can start to make some inferences as to what it is you're doing with light to try to treat the disease. Now, how do you get the drug there? So the question that comes up that I'm often asked is, it just doesn't make sense. You can't get light to the brain. The reality is that if you're thinking about visual light, that's likely true. In fact, it is true. You only get very small amount of light to the brain, not enough to create a beneficial effect in the neurons, but it is not true of near-infrared light. So I've done studies, other people have done studies, and what you find is that for the wavelengths of interest, in an adult head, about 5% of the light energy applied through the tissue, through the skin, gets to the cortical surface. That doesn't sound like a lot. In fact, it is a lot. It's more than enough. So one of the things I want to do in my talk is show you that, in fact, it does modulate brain physiology, and I get to that a little later. But another researcher that has done these studies measured it deep into the brain, and he showed Tedford that you can get light, near-infrared light, 40 or so millimeters into the brain. So detectable levels of near-infrared light. Without heroics, you're not going through photon counting measurements. Simple detectors, you can measure it that deep. Now, you don't really know that that amount of light is physiologically meaningful for a clinical effect, but it can be measured. So the next thing you have to be keenly aware of is that children have smaller heads, thinner skulls, thinner scalps, and some of the previous talks talked about the symmetry. The question is valid, right? We're talking about irradiance influence right away you realize that the dose for the child is going to be different than the dose for the adult. So part of my tools are modeling of light transport through tissue, so you can accurately model and predict how much light is going to get to the brain for a given wavelength, for a given irradiance. It turns out that just like you think, you get about twice as much light to the average 3-year-old than you would to the average adult. Well, you know, like with any other drug, you have to be keenly aware of that difference because, although it is plenty safe, there is an effect of inhibition. So if your dosing is not right, you put too much light into the brain, you cancel the effects. And that has to be considered when you're delivering and designing devices to use this technology as a therapeutic. So how does it work? So I'm sure here, plenty of you guys probably know more than I do. I spend a little time trying to understand what's going on, so I'll try to summarize my understanding of this behavior and years of trying to find out. The first thing you notice, or you got to be aware of, is that light interacts with tissue. We know this to be true. You don't have to go very far. You can go outside, get a tan. Maybe not this afternoon, but that's an interaction of light with your tissues. There are other endogenous chromophores in your tissues that when light interacts with them, they get absorbed, something happens. So just some classic examples. If you look at photosynthesis, the chromophore, the absorber is chlorophyll. It only absorbs in these wavelengths, around 500, which is yellow-green, and around 600, 700, which is red. Photosynthesis only happens at these wavelengths. So if I give light 550 nanometers, if I put 550 nanometer light on a plant, you're not going to get any photosynthesis. The same is true for color vision, where rhodopsin is a chromophore absorbing in the cones of your retinas, and you notice it doesn't absorb outside 700 nanometers. On the right here, north 700 nanometers, you have near infrared. And that's the reason why you cannot see near infrared. So when you're looking at a near infrared device for therapeutics, yeah, for all practical purposes, you see nothing. It looks like nothing is happening. That's important to know. Finally, for photobiomodulation, the chromophore interest here is cytochrome C oxidase. In the previous talk, it was mentioned that complex 4, cytochrome C oxidase, is involved in autism. One of the reasons, or may be involved, one of the reasons why this technology became apparently something you should try is because the mechanism by which it interacts with cells is well suited to the pathology of autism as it's becoming more and more understood. And interestingly, it absorbs pretty evenly in the near infrared. So that's another question that comes up. How do you know which wavelength to use, what color of light? Well, you pick lasers that fit, or LEDs that fit in these wavelengths, and there are thin enough tests in vitro to show that all of them produces responses in cytochrome C oxidase. So the wavelength itself is not such a critical choice, although you probably see that in terms of efficacy, 700 may be a little bit better than, 980, because the absorption by the chromophore is a little bit lower at 980 than it is 700. There are other things that come to play to the physics, but for the purpose of this introduction, all these wavelengths in the near infrared seem to have a similar effect. Now, what is it that happens? This has been tested in vitro. There's plenty of literature, so I'm not going to get substantially into it, but you can look at Bettini just last year. He did a beautiful job explaining how this may be happening to a cell. The first thing that happens that can be measured is an increase in the mitochondrial membrane potential. So that's necessary because that force drives the rest of the processes within the electron transfer chain. And it is followed by an increase in oxygen consumption and an increase of ATP production. Once that happens, you start to get secondary reactions that are a consequence of these increased metabolic activity, like reactive oxygen species, which lead to increased phagocytosis and decreased apoptosis. And you have secondary messengers that lead downstream to increase cell survival and decrease inflammation, and extracellular concentrations of ATP and calcium, which are intercellular messengers that start to create a biochemical reaction at the tissue level more than the cellular level. So at the higher level, the levels of the brain, what you start to see is a whole bunch of good things happen. So rather than getting through each one of those, I'm sure everybody can read them. This happens, this has been measured, this is being demonstrated. Here's another paper, recent, is the same Battini paper, where he discusses at length how this might be happening. I won't bore you with it, but if you're interested, this is a good reference. So, all these looks good, all this sounds good. Try it in vitro, it works nicely. And you have to ask yourself the question, is 5% or 10% of the light that you're putting at the skin enough to create a clinically beneficial response? Do you get enough light into the brain such that you can photobiomodulate its physiology to do something that you can measure at the people level, clinically? And the answer is yes. Here's a few references. So one of the things that we find is that cerebral oxygenation changes. We talked a little earlier about mitochondrial membrane potential. Yes, that goes up. Well, with that, oxygen consumption goes up, ATP production goes up. There are means to non-invasively measuring these things, and they have been done. And here's work by Tian that have shown that in fact this is happening inside the brain when you photobiomodulate from the outside using near-infrared light. And the two parameters we were talking about earlier in terms of dose, discussed a wavelength of 1064 nanometers and an irradiance at the skin of 250 milliwatts per square centimeter. So as a matter of reference, at about twice these, a person will start to feel heat. So below about 400 milliwatts per square centimeter, in this wavelength, you don't know that anything is happening physically. Now that's not true if you're using red light, because it's mostly absorbed in the skin. Much lower irradiances will be felt as heating the skin, so you will notice it. One of the beauties of using near-infrared light is not only that it is not visible, it is that at these levels, you cannot feel it. So blinding studies like these becomes relatively easy. What else is happening? Cerebral energetics. So we talked about this idea that you increase ATP production. Well, you do. You can do similarly and measure it at the organ level. This is a study using the same dosing, 250 milliwatts per square centimeter at the skin, 1064 nanometer laser, and they show that, well, in fact, cytochrome C was significantly oxidized, meaning it was donating electrons to the electron transfer chain so that the energetics of the brain cells were affected. What else? About blood flow. This is a study that I'm involved in, in which we're taking that technology and testing it to treat depression. What we found is that using BOLT, you can actually measure that the cerebral blood flow is changing. And in this particular study, the changes were dose dependent. I'll mention that a little later. And for this, the dosing was a little different. It was an 808 nanometer laser, which is still infrared, still invisible. It was a little higher dose, if you will, at the skin, but the effects were measurable and significant. And the reference is there for anybody that's interested later. I'm sure this is going to be published somewhere, available for everybody to look it over. Now, what else? What about EEG? I mean, we talked about these. A lot of the presentations are talking about EEG. And we see, just like before, there are changes that are induced by the light applied at the skin in the brain. So right away you start to look at it and say, okay, yes, there's target engagement. Yes, you can show multiple ways that you put light in the forehead at the skin and something happens to the brain. Now, I think this is important. The modulation of brain physiology is dose dependent. What do I mean by that? We talked a little earlier about two things that are important, wavelength and irradiance as determining your dose. Well, in this study, what we saw is that what we call a low dose, 808 nanometer light, 50 milliwatts per square centimeter, cerebral blood flow was decreased compared to sham. The change was statistically significant. If you try a different dose, what we call a medium dose, about 330 milliwatts per square centimeter, blood flow was increased compared to sham significantly. And there was, all right, you would think 12 years of electrical engineering would have helped me with these things, but okay. There was a third dose, which we called the high dose. The high dose did nothing. Now, when we went into this study, and you can call these bad research methodology, I would have sworn this would have been the best dose, period. So, a couple of things to notice. The irradiance is the same. These are no longer continuous, meaning the light is not applied, turned on, leave it on, turn it off. It's pulsing at 40 hertz. And this irradiance is average. So without getting into the details, there was a dose dependency that seemed to depend on the peak irradiance of the light applied. We're exploring that as we speak. And in multiple measures, this dose works best. One of the reasons I mentioned that it's important to know what's going on with autistic children and how much light you're going to put in, because, well, this is clearly not harmful. It also doesn't produce clinical beneficial effects. So, getting to autism, what I'm trying to communicate here. I think what I like to communicate is that JelikaLite has designed a technology, a dosing, that is based on real science and is designed for autistic children. So while PBM is getting a lot of attention and is being used for a lot of things, part of the problem what some of the studies failed is because the dosing is not understood well enough to be able to try it on everything and get a beneficial effect. So what is it about these devices that is important? First, it is actually stimulating areas of the brain that make sense. So for a neurologist, if you say we're treating the cerebellum, we're treating around the default mode network, we're treating around FP1 and FP3, and F3, oh yeah, it starts to make sense. There are 10 areas that are actually treated intentionally to try to produce some biological responses. It's 850 nanometer light, 40 hertz. So you ask yourself, what's wrong with you? You just told me 40 hertz wasn't working. Well, 40 hertz with this light for this pathology works great. It brings me back to this idea that dosing is absolutely critical. You have to do it right. They're designed for the children to wear. I mean, these are sensory children that have a problem with having a cap on their heads for 10 minutes at a time. So these things weigh 300 grams. They have no wires. You put them on the child. The child does whatever they need to do for somewhere between 5 and 7, 8 minutes, twice a week, you move on. Specifically to ASD. So some of the things we were talking about, photobiomodulation in the brain, how does it play into the ASD pathology? Restoration of mitochondrial function. That seems to come up over and over and over. There's a lot of data that shows that this is in fact happening. Increased neural connectivity, activity and connectivity, increased cerebral blood flow, reduced inflammation. There has been a lot of data collected, a lot of measurements taken, both in vivo, ex vivo, and in culture, that shows that some of the inflammatory markers, TNF, interleukins, they're all modulated in the right direction by this kind of light interaction. So one of the things that is sort of speculative, but it may be that one of the things you're seeing and the data is starting to suggest is that you're actually improving functional connectivity of the default network. Time will tell. So getting to the actual data. What happened when we put these devices together and we tested them? So first, some of the logistics. Our objective was to demonstrate that it was safe and effective, of course. And a secondary objective was to see what happened to EEG. Was there some kind of signal that you can use in the future, either as a diagnostic or as a tool to feedback tool to inform you of therapeutic effect? So we collected EEG over the period of the treatment, and the primary scale was CARS. CARS is, and most of you will know better, it is not really meant to look at differences. It's meant to be as an assessment tool, but in my understanding. But when you consider what is required to get an FDA clearance from the FDA, they think of CARS as a validated outcome measure. So the design of our devices has been given non-significant risk designation by the FDA and breakthrough device designation by the FDA. As part of that, it means that we can communicate with them in an open way, and in fact, they're kind of helping the process, and it accelerates the process of device development. But with that, the feedback becomes binding, and there's no negotiating as to what the primary outcome measure needs to be. It needs to be CARS. Practical or not, this needs to be what you have to modulate so that you can get an FDA clearance to ultimately take this technology to the children. Of course, IRB approval, informed consent, and at this point I should say this is not FDA cleared for treatment. We're testing it. So we have done 2 studies. First pilot study, RCT, 2 to 6 years old, 32 children were enrolled, 30 completed. The dosing was more or less the same between that study and the other one. 2 weeks dose titration. What does this mean? This is the first time this has been tested in this young children population. Trying to find out how it works, whether the dosing is too high, whether the dosing is too low. So we started at a low dose, and in this case, the way we titrated the dose is by changing the application time. So we started with 2 minutes. We see how the child responded. Then if everything seemed okay, you went to 4 minutes, 5 minutes, 6 minutes, 7 minutes. After 2 weeks, all the children were receiving the maximum time we felt comfortable with based on the energy deposited into the brain structure, which was 6 minutes. After that, they went for 8 weeks, 2 times a week. And during this time, there was EEG collected. There was a second study during which we wanted to test some non-dosing changes on the device design. This device was a little bit, it was homemade for all practical purposes. So we evolved that into a design that was a little bit more practical, functional, keeping the dosing identical. And we wanted to test these usability changes and also try to see what happens if we did things like increase the treatment time. In this case, one of the things that was important here was to expand the treatment population, the age, at least a little bit. And we wanted to include children from the first study sham group. So as a biomedical engineer, a physicist, to me, this starts to make sense, right? Sham control study, that's great. But if you can show that in the first sham control study it didn't work for the sham population, and you can take that population into an open label study, and at least show that it is working there, the dots will start to connect. So here are the results. Fundamentally, what we saw for CARS-2 is during our first study, RCT, there was a significant reduction in CARS of approximately 8 points. Now, this more than met the guidelines the FDA established for us as to what was a clinically meaningful significant change. They wanted to see 4 points. So we went to our second study and we reproduced what happened in this open label study. Remember, this population had children that were in the sham group on the first study. And we saw something very similar, about 7 points on average with a highly significant effect. This is starting to look good. Now we did take a look at EEG and trying to analyze EEG data as it's being presented. It's like trying to hold water in your hands. It's just, I looked at it, I did fast Fourier transforms, I looked everywhere you can think of. One of the things that made sense was to try to standardize these measurements in some way. And to my mind, one way to do that is to look at time. So this is the first EEG reading, and this is the last EEG reading, so it's from 0 to 1. We can do the same thing on the second set of samples, and we call that scale time. What happened is at the end of that, we saw that yes, the delta power decreased significantly in both cases. And that there was no delta power change in the sham group on the first. Now that starts to become interesting. I'll leave it up to the neurologists and researchers to try to interpret what this actually means. To me, it started to mean that you're having some anti-inflammatory effects and things in that direction. What we did, we also looked at theta. The first time we saw a change in theta, there was an increase. We saw not much the second time. We looked at gamma. We couldn't detect any gamma during the first study. There was a statistically significant increase in gamma on the second study. Are these artifacts of the small numbers, the technique being used for the measurements? Let you guys decide. Now, critically important, what's it say? Absolutely. Neither study showed any adverse events, no serious or moderate side effects. There were some mild side effects that were mostly about hyperactivity that resolved during the titration period of the dosing. So some children, as you increase the dose, they became hyperactive. Some reported headaches, but by the time we got to the main treatment protocol, they had resolved spontaneously. Now, just quickly for some conclusions, and this is an engineer speaking. Study 2. What was the goal of that study? The goal of that study was to reproduce study 1. Well, we did. The primary outcome measure looked beautiful. 7, 8 points change on the CARS, statistically significant in both cases. We reproduced the changes in delta power, which is an encouraging thing to look at. We found changes in gamma power we didn't see in study 1, and we did not reproduce the changes in theta power we saw in study 1. More to come. I think a much larger population, better methods, things learned, we're going to see that these are probably more reproducible than these things to show. And finally, again, an engineer talking, these changes in EEG spectra suggest a more alert, attentive, and focused mind. You can tell me if I'm misinterpreting these, but as I educate myself, trying to analyze and understand, this is what I get out of it. Next steps. So we got in here. We feel very good about the results. We feel very good about that technology, its safety, the devices, our interactions with the FDA. Things look great. So what are we doing next? We are doing a pivotal study. Unlike pharmacological applications of therapeutics, you don't have to do 2 or 3 trials and prove yourself right every time. For a device-based therapeutic, you have to have one trial, pivotal trial that shows effect. It has to be powered properly, and you have to have the right outcome. Everybody knows what's going on here. So, randomized double-blind sham control, 80 participants. This is powered to detect at 90% and 5% alpha. Male, females, 2 to 8 years old. Sunny in Mount Sinai. Basically, without getting into significant detail, moderate to moderately severe cases of autism. Some exclusion criteria, seizures, skin lesions, and psychotropic medication. Why? Just an abundance of caution. I mean, there is not a lot of data of this technology being used in seizures. Skin lesions can change the light transmission into the brain, so it'll be hard to say what the dosing is, and that's a variable you don't want to introduce in a clinical study. And psychotropic medications, some of the early work we were doing, we saw that some children that were not responding, when they were removed from their medication, they started to respond. So those are the reasons for those basics. Now, this is what the trial is going to look like. Screen, randomization, treated, sham, titration time, 8 weeks of treatment, a final reading. And what are we doing about here is our first site has already started. So as of last week, we started enrolling at Sunny. We expect that this is going to be finished sometime in June, 40 patients. We are going through the logistics with Mount Sinai and expect to start enrolling sometime in April, finish in September. Data analysis and review and FDA submission sometime early 2025. Now, just to give you some further background, if you go to clinicaltrials.gov and just start to mess around what else is going on with this technology, I looked at transcranial photobiomodulation therapy, and you find right away that there's at least 13 studies. Some of these is stuff I am doing for depression and Alzheimer's and Parkinson's, but we're not unique. We're unique in how we approach the therapy, the patient population we're treating, and how we develop the dosing to treat this particular indication. That's it. Thank you.
All right, we have questions for you, Luis. Dr. Adams?
Can you comment on how the dosing that you're providing equates to direct sunlight? Because it seems it's about the same level that you'd get on a sunny day.
It's actually quite different. The radiance of sunlight on a bright sunny day, it's 120 milliwatts per square centimeter. And of that, only about 1 or 2%, it's in the band of interest. So we're talking maybe 2 milliwatts per square centimeter compared to 150, 200, 250 milliwatts.
Well, actually, about half of sunlight is in the infrared. I mean, maybe not exactly at your frequency, but over the whole infrared, about half of it is infrared.
No, not exactly. You get about 35% or so is between 700 and 1100,
Okay.
but not all of those are going to do what we want to do. And some of those may be actually detrimental in that they are heating the tissue. So when you go out in sunlight, you don't only get 810 or 830 or 850, those wavelengths that might have a beneficial effect, but you also get 450 and 390 and 660. So you get things like additional heating of the skin, which brings vasodilation to the skin, which brings hemoglobin and deoxyhemoglobin, which prevents transmission of the near infrared. So at the end of the day, more of different wavelengths is not better.
Right.
So are you thinking of doing a mid-trial analysis? I know the Magnus Medical trial for TMS for depression, they did a mid-trial and the FDA stopped the trial because it was so effective they decided it was unethical to deliver the sham to the rest of the population.
That's a great question. I mean, to do an interim analysis, the short answer is no. And here's why. To do an interim analysis, you have to plan it a priori, so you have to have it in the protocol. And also there is a consequence to the statistical value that you need to reach to show efficacy. So if P of 0.05 is good enough for 80 patients and you do it at 40 as an interim analysis, it has to be P of 0.028, if I remember the number. It's not worth the risk.
On if biochemical or blood flow measures over the period of time that you give this stimulation or more afterwards, how long do the changes persist in any of those measures?
That's a great question. It's like turning the lights on and off.
Mm-hmm.
So when you're doing the bolt measurement, you actually see the way the protocol was developed for that study was I designed a device that you can bring inside the fMRI. You can wear it while you're treating, and the patient is unaware, the doctor is unaware, the rater is unaware, and for the first 10 minutes, there is no light, but you are recording. Then you turn on the light for 10 minutes, you continue to record, and you turn off the light for 10 minutes, you continue to record. So you have a 30-minute intervention. The middle 10 minutes actually have real intervention. And what tends to happen is within the resolution and the timing of the bolt signal, the minute you turn on the light, the signal starts to change, milliseconds. You turn out the light, and it almost immediately goes away. So for that particular signal, it's an on-off effect.
So that, and I think that could be good, right? Because one of the potential positive outcomes of this, like with hyperbaric oxygen therapy, is that it's not the oxygen, it's the response of genetic programs